All right, let's start talking about this series of reactions that makes up cellular respiration and oxidative phosphorylation. Let's talk about the actual series of reactions. I mentioned a minute ago that the process of glycolysis is involved. Glycolysis, or in some microbes, a slightly different but equivalent pathway, is going to be followed by a series of reactions called pyruvate processing, and that will be followed by a series of reactions called the Krebs cycle. Again, in some microbes, it won't be the Krebs cycle, but it'll be an equivalent pathway. Now, glycolysis plus pyruvate processing plus the Krebs cycle, those are all of the reactions necessary to break down all of the covalent bonds in glucose, or again, another simple sugar. And when all of those bonds are broken down and all of the electrons released, what we'll be left with is carbon dioxide. So from glucose to carbon dioxide, it's glycolysis, followed by pyruvate processing, followed by the Krebs cycle. Now, all of the electrons that get released from glucose during this process of cellular respiration, they have to go somewhere, and the place where they go is this structure called the electron transport chain. The electron transport chain is going to receive these electrons and literally pass them from one structure to another in this chain, harvesting the energy from those electrons as they're passed. The energy that gets harvested from all of those glucose electrons will be used to build that hydrogen ion gradient. The other term we use for hydrogen ion is just proton. So we can also call a hydrogen ion gradient a proton gradient. And when it comes to microbes, that's really the term you'll read more often. This gradient, this concentration gradient across the cell membrane, remember on one side there'll be a lower concentration of hydrogen ions, and on the other side there'll be a higher concentration of hydrogen ions. We give that a name in microbial cells because that gradient has energy associated with it. It has potential energy, stored energy. If we allow the gradient to resolve, if we allow those hydrogen ions to flow across the membrane down the concentration gradient, that energy can be used to do something. It can be used to do some work. And that work that's going to be performed is the production of ATP. Protons, or again, hydrogen ions, are going to flow from the place where there's a high concentration, and that's the periplasmic space, back across the membrane into the cytoplasm of the cell. Now, how are they going to get across the membrane? Remember, we said things that have a charge on them cannot cross between the phospholipids. Remember that? Things that have a charge on them cannot use simple diffusion to cross a membrane. They're going to need some help. They're going to need some facilitation. And the way they get across that membrane is by traveling through a tunnel in the center of a protein. And that protein is called ATP synthase. So when the energy from all the electrons that come out of glucose, when that energy is put to use, the cell is going to build a proton gradient, a hydrogen ion gradient, or what we commonly refer to as a proton motive force. Motive meaning motion, a motion force. The cell is going to pump hydrogen ions out of the cytoplasm and into the periplasmic space. Remember, that's the space between the membrane and the cell wall. So a big concentration is going to build up in the periplasmic space, and there'll be less and less and less hydrogen ion in the cytoplasm. Now we've got a gradient. Now we've got a difference in concentration from the outside of the cell to the inside of the cell. That gradient has power, it has potential energy, because as soon as it can, as soon as there's a way to get across the membrane, those ions are going to flow across. Nature doesn't like a gradient. Nature wants to get rid of that gradient. So those hydrogen ions are going to come back into the cell down the gradient by traveling through a literal tunnel in the middle of the ATP synthase molecule. And it's this motion of the hydrogen ions through the ATP synthase tunnel that's going to cause the ATP synthase to make ATP. The electrons themselves, the physical electron itself, still exists though. Remember, we took the energy out of them in the electron transport chain, but we still have the particles themselves to deal with. We can't forget about them. They need to go somewhere. And the place where they're going to go at the bottom of the electron transport chain is to that final electron acceptor. It might be oxygen, or it might be some other molecule. But the end of this long process of cellular respiration and oxidative phosphorylation is the acceptance of those electrons that came out of glucose by that final electron acceptor molecule. So I think it's probably clear to you why biochemistry, particularly metabolic biochemistry, is a course all of its own. You can spend all semester studying these processes of cellular respiration and oxidative phosphorylation because they're very complex. They have many, many steps in them and many intermediate molecules. But what matters to us in this course is not so much every single detail of all of these reactions, but what the big picture is about, why these reactions occur, and what's going into the reaction and what's coming out. In other words, what's the substrate that goes in to a process like glycolysis, and what's the product that comes out? What substrate goes into pyruvate processing? And what product comes out? What goes into the Krebs cycle? What comes out of the Krebs cycle? These are the things that are of primary interest to us in this course, and that's what we're going to talk about next. So, the processes of glycolysis, followed by pyruvate processing, followed by the Krebs cycle, are the combination that's going to break down a glucose, a 6-carbon molecule, to a carbon dioxide, which is a 1-carbon molecule. So we're going to get 6-carbon glucose broken down to 1-carbon carbon dioxide. And that breaking down is bond by bond by bond in a series of redox reactions. Remember, we said Leo goes grr, and that means to us that the molecule that loses electrons is getting oxidized, and the one that's gaining electrons is getting reduced. Glucose is what goes in at the start of all of this. Glucose is being oxidized to carbon dioxide. It is losing its bonds, so it's losing its electrons. That's the Leo. Now, those electrons have to go somewhere. Something else has to be reduced. And yes, at the very end of all of this metabolism, the final electron carrier is going to get reduced. But we're not ready for the final carrier yet. We need an intermediate carrier. And there are several of these electron carrier molecules. They're called things like NADH and FADH2. These are little intermediate molecules that are capable of receiving electrons from glucose. In other words, gaining electrons. There's your GR, becoming reduced. And they're going to then carry those electrons and pass them on to the next stage. They're going to give up those electrons that they're carrying when they transfer them into the electron transport chain. So those same carriers that got reduced when they gained electrons, they're going to get oxidized when they give those electrons up. They're going to give them up to the electron transport chain. The electron transport chain then is gaining them, so it's getting reduced. The electrons are going to be sent down this transport chain. And I know that language seems very strange, but the purpose of the electron transport chain is to harvest energy out of these electrons. So the electron transport chain will be oxidized for the final process as the final electron acceptor molecule gets reduced. So from the beginning of cellular respiration to the end, we're going to see oxidation, reduction, oxidation, reduction, oxidation, reduction, oxidation, reduction. It all begins when the first bond in glucose gets broken, and it ends when the final electron acceptor molecule gets reduced. So, glycolysis, pyruvate processing, the Krebs cycle, followed by the electron transport chain, followed by what? Well, all of the energy that gets harvested from those glucose electrons, all of that energy is going to be used. All of that energy is going to be put to some use so that we can make ATP. The electron transport chain, when you look at its structure, is simply a series of proteins that are embedded in the cell membrane. These are kind of special proteins because they have these additional sort of small molecules that are added onto them that give them very interesting electronegativity properties. What that means is that these proteins that make up the electron transport chain are really good at moving electrons and harvesting the energy out of them. I like to imagine the electron transport chain like a staircase. When you stand at the top of a staircase, just like if you stood at the top of a mountain, you have lots of potential energy in that moment. Now, what do I mean by that? I mean that if you were to fall, if you were to fall down the steps, or you were to fall down the mountain, all of that energy would be released. And by the bottom of the mountain or the bottom of the staircase, you wouldn't have a lot of potential energy anymore because it would have been released into motion, into kinetic movement as you fell. So in come these electrons at the top of the staircase called the electron transport chain. These electrons are not going to fall down the stairs. These electrons are going to walk very carefully down the stairs, one step after another after another. They're going to get lower and lower and lower down the staircase. They're going to be reduced in their energy as they go step to step to step. And the energy is going to be harvested by this transport chain. By the bottom of the staircase, those electron particles have less energy than they did when they were at the top of the staircase. And the energy that they have lost has been taken by those transport chain proteins. And it's going to be used now to build that ion gradient, that hydrogen ion gradient. At the bottom of the staircase, the electrons still have to go somewhere. We can't just leave them at the bottom of the electron transport chain. They have to go somewhere. They have to be received by a molecule. In other words, that molecule has to be reduced. And that's where oxygen comes in, or some other final electron acceptor. Now we're building a proton gradient across the cell membrane. The energy that was harvested from the electrons as they pass down the electron transport chain, that energy is going to be used to pump hydrogen ions from the cytoplasm across the membrane into the periplasmic space. Remember, we talked about active transport. We said pump proteins do that work. They move molecules against their gradient, up the concentration gradient, from the place where there's a low concentration to the place where there's a high concentration. Active transport. You have to fuel it. And we're going to fuel it with all the energy that we just collected in the electron transport chain. Protons or hydrogen ions will be pumped out of the microbial cell, across its membrane, and into the periplasmic space until we have a nice big gradient built up. Nature doesn't like that gradient. Nature wants that gradient to go away and go back to equilibrium. And that's going to happen. Those hydrogen ions, those protons, are going to be allowed to flow right back into the cytoplasm where they belong. And they're going to do that by flowing through this molecule called ATP synthase. This molecule has a physical, literal tunnel through it. And as those hydrogen ions pass through that tunnel back into the cytoplasm, their motion, their kinetic energy is going to be transformed into chemical energy in the form of ATP. On this slide, you can see a piece of membrane that came from a microbial cell. This is the area that represents the cytoplasm. Here's the cell membrane. And this out here represents the periplasmic space. So the cell is going to pump hydrogen ions out into the periplasmic space and build up a proton gradient, a hydrogen ion gradient. This funny looking structure right here in yellow and orange, this is ATP synthase. We know the physical three-dimensional structure of ATP synthase. We know what it looks like. And part of it, the yellow part, is actually embedded in the cell membrane. This is the part that has a tunnel through it. The other part of the molecule, what looks like a pumpkin hanging into the cytoplasm, this part is actually movable. It's hanging off of this stalk, as we call it. This thing, this pumpkin-looking portion of the molecule, can actually rotate. It can actually turn around and around on that stalk. So hydrogen ions, which build up out here in the periplasmic space, they're going to flow back in through the yellow part of the ATP synthase and into the cytoplasm again. Their motion, their physical motion, has energy associated with it. That is kinetic energy. This molecule is going to transform that kinetic energy into a different type of energy. And that different type is stored chemical energy. It's going to take ADP and phosphate and that energy, and it's going to build ATP. This is what the first law of thermodynamics is about. You can't make energy out of thin air. You can't destroy energy. But you can take energy of one type, motion, kinetic energy, and you can transform it into another type, chemical, stored, potential energy. You should know that every cell on our planet has this molecule in it. It doesn't matter if you're talking about a human being or your dog or the plants in your garden. It doesn't matter if you're talking about a bacterial cell or a fungal cell. All of the cells on this planet contain ATP synthase. Isn't that remarkable?